I'm now going to talk about what happens for patient samples in the laboratory when they have or they're going to be diagnosed with chronic myeloid leukemia. I'll reference back to some of the earlier videos where I talk about the biology of CML, and even back to the even earlier one where I talk about sort of some basic biology for the CML patient. But the journey starts for most CML patients with a blood test, and that can be done for a variety of reasons, because you're symptomatic, for just a random check, and it's often done by non-haematologists, GPs, accident emergency, that sort of thing. And we get some clues as to whether you've got chronic myeloid leukemia based on the numbers in a full blood count. So that looks at your hemoglobin, your platelets, but for CML specifically your white blood cells, and a little bit about what white blood cells there are present in your blood. And often in CML you have far too many of them. So once we've got those blood counts, which are provided by the automated analyzers, someone will have a look at the blood down the microscope, because it'll be probably very abnormal. And when you look at the blood down the microscope, like this picture here, you'll find far too many white blood cells, which are sort of blue and purple blobs here. But the makeup of those white blood cells can give us a really good clue that something like chronic myeloid leukemia is lurking. Things like having too many basophils or eosinophils are a good clue. And patients, when they're diagnosed, can have anemia. They can have high or low platelet counts, as well as having usually a high white blood cell count. So the next steps are, we now need to make a diagnosis of chronic myeloid leukemia. And as I've talked about in the biology of CML, the key defining feature is the BCR-ABL fusion gene. So where the BCR and ABL1 genes get stuck together. And there are different ways we can detect that. We can look at the chromosomes under the microscope, but sometimes the quickest and the easiest to explain way is something called fluorescence in situ hybridization. I'm sure you can look that up in detail if you want to know more, but very simply, we have these sort of colored probes which stick to the right genes on the chromosomes, on the DNA. So we have a green probe, which will be to ABL1 on chromosome 22, and a red probe, which will stick to BCR on chromosome 9. And in a normal, healthy individual, these two probes, when you look at these cells on the microscope, will be nicely separated. But as you can see in this picture here, if the green and the red sort of light gets put together, that means the two genes for which these probes have stuck to have become stuck together. And this is an example of a positive, what we call FISH test, to demonstrate this patient has a BCR-ABL fusion. So that's really important. There are different ways to do it, as I said, but we have to demonstrate there's a BCR-ABL fusion. And then what a patient will need to undergo is probably a bone marrow biopsy. They might have had that before we've decided they've definitely got CML. Sometimes we will need to do it anyway, but I'm sure many of you who are already CML patients will have experienced this. Not a lot of fun to go through, but really essential to make sure that we're in the right stage of the disease. We're in chronic phase rather than in a blast phase, and looking for other abnormalities in the chromosomes of your CML. So also a really important thing to do for almost all patients. So that's how we diagnose CML. Bone marrow biopsy, and then demonstrating there is a BCR-ABL fusion. So, and then patients are given treatment, etc., etc. And then the next stage is, how do we monitor the CML? And really, again, a very useful fact about chronic myeloid leukemia is that not only can you have a targeted treatment, such as your TKIs, to treat it, but you can also utilize the fact that there's a BCR-ABL fusion to monitor. And monitoring the presence of that is very useful to monitor the disease. How do we do it? Well, it's based on something called a polymerase chain reaction. So you might be referencing something called PCR that might have come up in letters or things that your doctors say to you. So polymerase chain reaction is just a way that we replicate DNA in the laboratory. Basically saying, I've got a couple of copies of DNA, I'm going to make loads more so I can then measure it or detect abnormalities in it. And basically we do that by getting some DNA, heating it and cooling it a bit, and then adding other things to the reaction to just grow more DNA. Again, if you want more detail on that, there'll be plenty more information out there in nice videos explaining polymerase chain reaction. Crucially, in chronic myeloid leukemia, we need to be monitoring the RNA. So as we talked about in an earlier talk, the recipe book is your DNA, but actually it's very, very hard to turn that into a good laboratory test, which accurately measures the amount of BCR-ABL there is. Again, it's never going to give you an accurate result. So what we have to do is we have to utilize the mRNA, the photocopied version of the recipe book, and say, actually, that's how we're going to get an accurate measure of how much there is, because we've got rid of all the other stuff that's in the DNA, the bits of DNA that aren't coding for the protein. We've got rid of all that, and therefore we can accurately monitor it. The crucial thing is that RNA that we're measuring is actually only stable in these cells for a limited amount of time. So even after about 24 hours, it's starting to degrade and break up. And by 72 hours, it's broken up so much that we can't accurately measure it. So what we always ask for, and this is why as a patient, if you've been asked, don't take your samples on a Thursday or Friday, it's because that RNA doesn't last long enough to get through the weekend if the laboratory is shut. So that's really important. But once you've done that, what we do is we get that RNA and we turn it back into DNA. So we re-photocopy it into DNA, which is much more stable. And then we run what we call a RT-qPCR, which is essentially a way of monitoring very accurately the amount of BCR-ABL. And I don't want to go into too much detail here, but essentially it's the same as the polymerase chain reaction we've just talked about. But what we do is we add in what we call a probe that will be released whenever the polymerase chain reaction happens, and that emits light, and that happens as the product grows and is in relation to the amount of BCR-ABL there is, and that therefore the amount of light is how much BCR-ABL there is, and we do some funny stuff in the lab to link it all together, and that gives you a percent of effectively the cells that have BCR-ABL and gives you an accurate result. And then the laboratory turns that into an international standard result so that you can hopefully replicate that across laboratories. And that's a really important bit of CML treatment, is accurate monitoring.